Andreas, I think that the floor is yours.
Welcome everybody back. I'm happy to share the next session, and I'm very happy and delighted to have Professor Paula Giunti. She's a very known speaker on every ATAXIA conference, and Helen is a Principal Clinical Research Associate in the London Research Center, and she is one of that person who established in 2005 the first ATAXIA center in UK, which is also rated by ATAXIA UK as an excellent center. So I'm very happy that you're here.
Thank you, Andreas. And Kalimera is the only Greek word that I know, so I will carry on in English, obviously. And so thank you so much for inviting me, and it's really, really exciting to be with the organization and with the patients, ATAXIA, and the local Hellenic ATAXIA group organization. So I'm going to talk about biomarkers in spinocerebellar ataxias, and it was very good that I came after the speech of Jörg that very clearly really pinpoint the relevance of biomarkers in the work, research of spinocerebellar ataxias, including Friedreich, of course. And so biomarkers is a defined characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or response to an exposure or intervention. So we have several biomarkers for diagnostic, for progression, for safety, toxicologies, and so forth. Today I'm going to focus on the fluid biomarkers, biomarkers that can be very useful to track the evolution of ataxia patients and could be endpoints for future clinical trials. So I focus, and my group has focused on 4 analytes, so the NFL, neurofilament light chain, and the GFAP, fibrillary acidic protein, tau, and ubiquitin carboxyl terminal hydroxylate L1. Why? Because I thought that these were very good proteins that are involved in neurodegenerative process and neuroinflammation process. We know that now that the majority of the spinocerebellar ataxias has both components. And these are obviously damaging the nerves. And what I wanted to show that so what I wanted to show is that always, unfortunately, it's not very clear here and very faint. But anyway, the GFAP is coming from the astrocytes, whereas the tau dendrite, and then the neurofilament from the axons, and then the UCLH1 from the body and the cytoplasm. So we it's essential for the biomarkers and for rare diseases to have a very large cohort of patients because of the variability. And so we are part of the European for spinocerebellar ataxia, Mageddorf-Joseph disease consortium led by Professor Glockater, but also we really collaborate very tightly with the Mayo Clinic, Professor Len Petruccelli, and Professor Prudentius. And this is what we intended to do, and with the 2 courts, the US court and as well the court for Europe. So it has been really relevant to have a court in the US that was a very different background, ethnicity, and so forth, and compare and to validate the analyte for the biomarkers for the study in spinocerebellar ataxias. So what we concentrate on the plasma and CSF. And I would like to highlight in this talk really the relevance of having CSF from patients. And the CSF has been always really seen as very, very invasive, and it is. However, in our hand, in the expert hand, this had never had any major side effect. So this is what I wanted to say as well for the patients. So the analytes were then recorded, and then the test for the plasma and the CSF. But then what is really important is to correlate the level of the biomarkers to clinical assessment and to clinical like SARA, INAS, other biomarkers, ADL, and others. So we concentrate on the neurofilament, and neurofilament has been already seen being relevant in other neurodegenerative diseases like Huntington disease, but also in other conditions. There are neuroinflammation is the main primary cause, trauma, and so we wanted to as well validate it in spinocerebellar ataxias. And the neurofilament has a different component, and these are the neurofilament high and then medium and light. So there are two components, one coming from the CNS, so the central nervous system, and the peripheral from the peripheral systems. So this was the code that we share with the Mayo Clinic, so the Mayo code and the UCL code. And the most important things that this is really busy slides. The only reason why I put it there is to convey another very important message, that the code needs to be homogeneous. So these were indeed homogeneous for the age at onset, for gender, and for the SARA score, and then the number of CAG repeat. So with this in mind, and having set up the scene with a very strict criteria, we went on and we looked at the CSF of the two codes. And indeed, as I hope, yes, you can see, this is the healthy control, and then this is the NFL from the SCA3, and then these are the pre-symptomatic and the other spinocerebellar ataxias. So there was clearly a very high NFL on the CNS when we look at the liquor. And what I wanted to highlight here is that obviously the other spinocerebellar ataxias were heterogeneous, were SCA 1, 2, 3, 6, and 2 and 6. And as you can imagine, there is a difference. So SCA3 was very high, and we would expect to have other spinocerebellar ataxias that were much less on an NFL. So despite you have biomarkers that can serve one condition, they may not as well is relevant in other conditions. And this is something that we need to bear in mind when we are treating and when we design clinical trials for the spinocerebellar ataxias, as these are rare diseases. And the majority of the patients always asking me, but why don't we put everyone together? Because they share the molecular substrate, they share the clinical features, and some of them the progression. But sometimes this is not possible, and this is one of the examples. Then we looked at the plasma, And again, we have this increase. This was replicated in the the validation code, main code. So we have a very similar, in very similar fashion, they increase. And what is interesting is that in the pre-symptomatic, where you don't have sign of the conditions, then you have a lower NFL. Looking at the specificity and sensitivity, these are pretty good, and it's really incredible. You would see is 1 when we do the ROC test for the CSF, whereas less for in the plasma in the main code. And in the validation code, similarly. So this is again, another very busy table, and what we wanted to do is to correlate the NFL to all the other parameters and the clinical parameter, and so to see that there was perhaps a correlation. And unfortunately, there was not a very tight correlation with these parameters, clinical parameters. So we thought maybe we would have done something wrong, and we went back and we just went on and looked at one of a target engagement parameter, that it is the CAG repeat. And indeed, when we did the multivariate analysis, the correlation was really, really still high. And so with a very high p-value, statistical analysis was very sound. So possibly there was an issue in being this code too homogeneous, possibly. And the other things to note is that there was a correlation, a trend of correlation on SARA, and in the other scale, the Mayo Clinic has thought about the gait mobility score. So there was a trend, but possibly the fact that it was too low numbers then was not full statistical significant. So what we did at UCL is to have a higher number of biomarkers, a higher number of patients. And so we collected all the plasma and for the ESMI code. And so we concentrate on the plasma with the Simoa, who is capable to read very, very, very microscopic level of these analytes. And we use a kit where NFL, t-tau, GFAP, UCLH1 were encompassed. And then for the CSF, slightly different assays. So the ELISA for the NFL, and then the INO test for the tau, and INO test phospho-tau. So we arrive with the ESMI code, 172 samples, and then the validation code was, in our hands, the Mayo code, and always the same 54 patients. So the tau plasma levels in SCA3, we focus on the tau, and very interestingly, we saw, and surprisingly, we saw that tau increases in the pre-ataxic, whereas decreases in the ataxic. And this was something that it was very, very intriguing. They then replicate this one as well in the plasma and then in the CSF. This is the data just plotted on the graphs, and you can see that the pre-ataxic exceed the ataxic one. So there was as well the correlation between the tau and the INAS. So I would like to remind everyone that the INAS is the inventory of non-ataxia symptoms and signs, and this is not a scale, but it's just an inventory. So it gave you the gestalt of the patients. It gave you the understanding how many extra cerebellar signs there are, the complexity, because we know that the spinocerebellar ataxias are very complex. So yes, there is a cerebellar component, but there is a peripheral component and extrapyramidal features and so forth. And this was as well interesting. And then we looked at the CSF, and again we have this the pre-ataxic, they were much higher than the ataxic one. And the same we correlate with the disease duration, and there was the trend as the disease duration progressed that the tau decreases. So this was very intriguing, but we couldn't go further. We couldn't increase the CSF chord because, as I said at the beginning, for patients it's very difficult to accept the lumbar puncture and so forth, and it also required as well an admission to the hospital. So I called my friend and colleague Luis Pereira on the University of Coimbra, who has mice, transgenic mice, for the Machado-Joseph disease, the SCA3, and I designed for him a protocol just to look at the tau in his mice and looking at pre-ataxic stage and post-ataxic stage. And he could demonstrate clearly that when the mice is 4 for 5 weeks, there was no ataxia, you have an increase of tau, and after, when they become symptomatic, the tau decreases, mirroring completely what we have observed in humans and in patients. So the plasma NFL, now we moved on NFL. We wanted to do validation. We have done previously with the Mayo Clinic, but now we want to do as well to see if our kit was able, because it's a slightly different assay, and so that was as well the aim. They replicate completely what we have found with the ELISA. So we have the control, and then as the pre-ataxic stage advance and the ataxic stage advance, we see an increase of the NFL. And this is plotted differently, and this is very clearly that in the pre-ataxic stage, patients has less level of NFL compared to the ataxic that are in blue. So again, the rock showed very good AOC, 0.9, and we wanted to correlate the NFL, and this time with a higher number of patients, we indeed found that there was a correlation with the SARA score. That was absolutely reassuring. So in conclusion, the t-tau level are higher in pre-symptomatic younger patients, and compared to control, and they tau correlate with the INAS. The plasma NFL levels were higher in mutation carrier compared to controlled, and this was again seen by the Chinese group, by the German group, and others. And then there seems to be that SARA was the stronger predictor of NFL in ataxia patients. And unfortunately, the GFAP and the UCHL1 did not show any difference, and there was non-contributory on this for identified new analyze new biomarkers. So I would like to thank, obviously, the whole work has been done by my PhD student, Hector Garcia Moreno, and obviously at the Mayo Clinic, Professor Len Petruccelli, and especially Professor Prudencio. And I would like to thank the ESME Consortium, and the ESME Consortium is ongoing. And so that's, it's very, very good that we could manage as well to attract funding to carry on the natural history for that. And, you know, a big, big, big thanks, obviously, to all my patients. Thank you very much.
Well, our next speaker, I'm very happy. She worked since 1988 on FA and built up her own team in 2001, as I researched it correctly, in INSERM. And I'm happy to have Dr. Helena Puccio here, who is talking about the Treat ARCA project, which the ATAXIA is also involved in. As well as the German association, and supporting this enormous, important project. Thank you.
So I'd first like to thank both ATAXIA and the Hellenica FA Association for having me present the work. And so I thought I would do two aspects. First, give you a little bit about how using animal models and cell models can be important to understand both disease and test therapeutic approaches for ataxia, and also then give you a little update on what is Treat ARCA, which is a European consortium. For development of new treatment, either for both autosomal recessive ataxia, which is what we call ARCA, versus the dominant ataxias, which tend to be called SCA, and it's the same thing for both diseases, both type of diseases, but we need to develop disease model that closely mimic the human condition, because, you know, we can explore disease models in many different ways, which we cannot explore human. I mean, the humans can be explored by cohorts, but they have their limitation. And I will make an emphasis on how important it is to get good disease models, and maybe we can talk about this after. Disease models enables us to develop preclinical proof of principle studies, both for repurposing drugs, small molecules, new molecules, and gene therapies. And of course, we can also use, we just heard about biomarkers, but we could also use good disease models to try to come close to biomarkers, or to validate some of the biomarkers. So there's research in my group. You know, our basic research has been to try to understand disease mechanism to be able to develop a therapeutic approach. And as Andrea said, I've been working on FA since 1998. So Friedreich's ataxia is due to a loss of function of the frataxin gene. And this frataxin gene is very important for what we call the iron sulfur cluster metabolism, which is something very important for the mitochondria. I've also been working since 2008 on another rare disease called ARCA2, or COC8A ataxia. This is a very rare, ultra-rare ataxia. There's about 100 patients worldwide. But it's also involved in CoQ10 metabolism, so it's also part of the mitochondria, and so it's very important for mitochondria also. So I thought that looking at these two ataxias might help us better understand. Just very quickly, what are the mitochondria? Some of you might know, but some of you might not know. These are the powerhouse of the cell. So this is where actually all the energy necessary for the cell comes from the mitochondria, or most of it comes from the mitochondria. But they're also extremely important for all the good metabolism of most cells. And so mitochondria are extremely central to the normal functioning of a cell. And so you can imagine if we have proteins that are involved in mitochondrial function that are mutated in ataxias, I mean, this will lead to dysfunction of many different cell types. And the organs that need high energy, for example, nervous system, the skeletal muscle, or cardiac muscle, are particularly vulnerable to mitochondrial dysfunction. And this is why in some of these mitochondrial ataxias, you're getting vast type of symptoms, including not only ataxia symptoms, but you're also getting cardiomyopathy, for example, in FA, or skeletal muscle myopathy in ARCA too. So my lab has been working on really concentrating on generating cellular and mouse models and zebrafish models to understand both what is going on in the disease, the physiopathological mechanism, but to drive to therapeutic approach. What are the different type of models that we develop in the team? So we have both cell models. So what is a model first? A model is something that needs to reproduce some aspect of the disease, and you will see there's no perfect model. And this is why, and I know it's often confusing, this is why there are so many different type of models that we have, and that there's not one model that's better than the other. They're all complementary models because they can't answer everything. We cannot reproduce the human disease in a model system. So we're reproducing parts of aspects of the disease. So we have cell models, so we can have cells derived from patients, and this is why it's important to be able to do biobanks. So we can get PBMCs or fibroblasts, but often these models don't have the phenotype they don't have the disease. They express the genetics part, but they don't express the disease parts. The recent advances to what we called induced pluripotent stem cells, or IPS, I'm sure that many of you have heard about this. This is being able to reprogram to take from either PBMCs or fibroblasts to be able to go back to a stem cell-like phenotype, and then in vitro, using a combination of factors, we can theoretically differentiate these in many different cell types and make little organs. And actually here I'm showing you what we're doing right now is we're doing a dorsal reganglia organoids. I don't know if you can see this, but this is equivalent of a dorsal reganglia, which is all along the spinal cord. And so this is one of the organs that degenerates in FA, and we can recreate an organ that has both neurons but also have glial cells. And so we can try to come closer to the human system, and this is of course based on human cells. And so these are important to be able to reproduce, but they're not perfect. They're very immature type of organs. And then of course we have primary cells derived from animal models, and here you can see an example of a Purkinje neuron derived from an animal model, and the Purkinje neurons are the ones that are primarily affected in a lot of the spinocerebellar ataxias. And then we have animal models. We have the mouse models. We can genetically modify the mouse models to try to make the same mutation or to mimic the effect of the mutation in the mouse. And I will encourage, if something that the patient association can do to help the researchers, is that Europe is tending towards wanting us not to use animal models anymore. And this is absolutely extremely important for us to be able to carry out work on animal models, to do preclinical work. And so this is where you can also help us, because we need to be able to defend the use of animal models, of course, under good ethical conditions, and this is what we do. I mean, we have to fill out a whole bunch of ethical, to work on a mouse, it's as much ethical issues as to work on a human. But this is something very important. And then more recently, we're also developing a lot of zebrafish models, and this is because we can genetically modify zebrafish, so these are little fish. We don't have as much ethical difficulties with Europe with the zebrafish, and zebrafish can also be used to do some drug screening. So I'm going to give you a few stories just to show you what we've been doing in the lab. I want to talk about the gene therapy effort because I think this is something that is of interest to a lot of people. I want to remind you that what gene therapy is. So it's the introduction of genetic material to the cells to correct the effect of the defective gene. And what is used very often now in clinic as a cargo, right, or in preclinical development as to deliver the cargo are these adeno-associated viruses, or AAVs. And this is being able to deliver the gene to the cells. And the reason it's able to do this is because this is based on what a virus usually does. So this is a very extremely simplified little scheme, but to help you understand, a normal virus will infect a cell, so the virus knows how to infect the cell. It will come into the cell, deliver its DNA, if it's a DNA-based virus, into the nucleus, and then it will be able to hitchhack the whole cell system to replicate itself, make more viruses, and then come out and then infect other cells. So that's normal. So we're using, for gene therapy, we're using the capacity of the virus to infect cells, to deliver DNA. But of course, we're making a disactivated virus that has the genetic material of interest. So here I'm showing the scheme with frataxin, but you can imagine any genetic material that you can put. So we put frataxin instead of the viral genome, we put frataxin in. We pack it up in a virus. We keep the capacity to infect the cells, and then it will deliver frataxin into the nucleus, and then the cell will make frataxin, which will go into the mitochondria, and then that's a way to do gene therapy. So that's the basis of gene therapy, at least in a disease that's where there's a loss of function of the protein. There's other types of gene therapy in gain of function. So we developed a gene therapy approach using our conditional mouse models. So these are mouse models that have organ-specific deletion of frataxin. So these are FA mouse model. We have both a cardiac model and we also have a neurological model. And these models actually have been the basis of a lot of research that we've done in my lab that's been able to help us understand the function of frataxin, along with other labs working on that, and allow us to understand the cascade of events when we don't have frataxin, and to test therapeutic approach, both pharmacological and gene therapy. So we did the first proof of principle that gene therapy could work for prevention and reversal of the cardiac phenotype in FA. We had done an IV injection of a AAV, so that little virus that expresses human frataxin, and we were able to show that it could not only prevent the onset of the disease, but reverse the onset of the disease in a mouse model. And that enabled us to also show that there was a large therapeutic window. We further went on and developed some dose response study, which allowed us to show, to see how much percentage of the cardiac cells we need to be able to treat. And we actually showed that only about 50% of the cells need to be treated in order to be to have a functional heart. And this is very encouraging because we know that in the human, we'll never go to 100% transduction. So we won't never infect all the different cells. We also showed that overexpression of frataxin, as Marion said, has a toxicity effect, and so this has to be very careful. And so this is one of the events that actually slowed down a lot of some of the development in clinic also. And then in collaborations with pharma, there were studies in large animals for delivery methods. As Miriam Rey just said, we're happy to know that based on our original data, that there's a phase 1 clinical trial launch for the gene therapy by Lexeo and the Cornell University, and so we'll see what happens. We also very quickly after did the same proof of principle for the neurological aspects, and we were able to also show that there was a complete reversal of the sensory ataxia linked to therapy. And because I always need to show little mouse videos, I hope this is going to work. Okay, I do this. So this is to give you examples of what we do in labs. So this is a normal mice walking on the bar. So this is our way to look at ataxia for a mouse. Here is an ataxic mice, I hope you can see, that is not treated. So you can see that it's having a hard time walking on this bar. You can notice it's using its tail to kind of balance itself and all that. And so this is a pre-symptomatic treatment. So this is before the onset. And so here's the little sister that has been treated. And you can see that it absolutely did not get the disease. So this is how we can show prevention of the disease. And then I'm going to show you a second slide where now we're coming and we're treating post-symptomatic. So the mice is already sick. And so what I'm going to show you is a mouse at 8 weeks that is not treated, and its little sister that was treated 1 week before. So when it already had the disease. And you'll see it's not perfect, but it's getting better. And so here's and it's going to take some time to move on. So on the left, you have the non-treated animal. And so you see it's really having a hard time going, and this is it's a more difficult, this is maybe like the equivalent of the peg board or something that you guys might have seen in clinic. So it's having a really hard time going through, and you can see that the treated mice, I'm just going to put it back, it's not walking perfectly over this obstacle, but it's still walking better. And so these are ways for us to look at gene therapy or any type of therapeutic approaches. So that's why mice are important. And so then we can, of course, look at these treatment, and we can look at them, can't see much, but you can look at them where they're already sick when they have, when we're treating them, and then we can correct, and you just cannot see the dark gray, which is the corrected one. That's too bad. And similarly, we can, with other different tests, we can show a complete correction of the disease. So where are we on gene therapy for FA? The ongoing research in my lab directly, but I know that other labs are doing also that, and some pharma are doing that, is dose-response study, trying to see how much neurons we need to correct. We're testing other AAVs to have the blood-brain barrier, to be able to pass the blood-brain barrier, because we need to pass that to go to the cerebellum. And we're testing frataxin mini promoters, so little ways to change the expression level of frataxin, because I said that too much frataxin we know is toxic. So we want to go to a level that's very close to endogenous, so to the normal level of frataxin in the cell. And so we're trying to use the normal elements that regulate the expression of the gene in the frataxin genes. And so we're playing around with that. As Miriam said, there's many pharma and academic science that are developing gene therapy for the neurological aspects of FA. And then we're all dealing with the same type of issues, the issues with delivery of the vector, because we need to target all the cells, issues with toxicity of the vectors, issues in manufacturing of the vectors, and this is not this is a big issue, and issues in clinical trial design. And this has actually been very important for where the patient associations have been very important to helping farmers doing that. So that was a quick part on gene therapy. I don't even know how much I'm talking, but the second part I wanted to show was maybe how we can use cell models to come in and find new therapeutic approach. And so here, this is the pipeline of what happens when frataxin is depletion. We know we have different aspects, and when the pharmacological approaches are hitting at different parts. So we can either try to upregulate frataxin, as Jörg Schulz said, or we can come and go on the symptomatic part, and for example, the NFR2 activators actually are the symptomatic part. So we had a question is, where can we bypass frataxin? So if we don't have frataxin, can we find a way to bypass frataxin? So again, with a small molecule approach. And so what we did is we put cellular models. So we took dorsal ganglia, which are the cells that are affected, or the proprioceptive neurons, sensory neurons, which are the cells that are affected in FA, from a mouse. And so this is a spinal cord of a mouse embryo, and we can see all these little balls. We take all these little balls, we dissociate them, and we put cells in culture. And so here you have the neurons in culture. We can see the neurons are expressing neurofilaments. And you can see that these neurons don't have any frataxin in them. So we have neurons with no frataxin that are live in culture. And so what we can do, and then we look at these neurons, and we can look at the phenotype. And these neurons actually have many, many different phenotypes. So they have many phenotypes that look like FA phenotype. But they have a mitochondrial dysfunction. So you can see here, in blue are the neurons that are affected. You can see that their mitochondrial function is less than in control neurons, and then if we bring frataxin back, we can rescue that. And then you can also see oxidative stress. So this is when mitochondria is not working well, we have oxidative stress, and you can see that there's an increase in mitochondrial oxidative stress in these neurons, and then when we bring frataxin back, we can rescue that. And then the neurons also are smaller. You can see here the control neurons and the frataxin deficient neurons, and they're actually smaller, and when we bring back frataxin, they get back to normal size. And so we decided to use this to do a pharmacological screen. And so to do a pharmacological screen, we need to have a lot of different steps. I'm not going to go through all the different steps, but one of the biggest important steps is to establish a protocol to do this in small little plates and to be reproducible. And so this is what Olivier Grisot did in my lab. So he set up a whole pipeline to do high throughput screening, where he's able to do this in these small little plates. You can see here 96 well plates, and then he can optimize many different things, and then this is all done under an automated robot so that it's as reproducible as possible. And by doing this, he was able to screen 1,000 molecules and these are FDA-approved compounds, so this is what we call repurposing. So it's compounds that have already been accepted for something else, and we're seeing whether they can work in another disease. Out of those 1,000 molecules, there was quite a few molecules that had showed some type of effect, either on the size or on the oxidative stress, or on both. And so then we went on and validated 48 molecules in a dose response manner. And so here's just an example of what it looks like as a readout. We have molecules that increase the size, so all the ones that are above this line increase the size of the cells. And here we have molecules that reduce the stress of the cells. And so you can see some compounds are doing on both, and some compounds are just doing on one or the other. And so that's where we are with that. And so we're able to do a high throughput screening. We have identified 48 compounds, and out of those 48 compounds, 18 of them are look to have some type of dose response. And so now we're trying to further develop these drugs and to see if they have an effect in animal models. And if we still have 5 minutes, 2 minutes, I don't know. Yes, okay. So this is to show you why it's important to have good models, both mouse models and cell models, in developing treatment approaches that then hopefully can go to humans. The last part is I want to present to you a European joint program that we have built a few years ago to try to develop treatment for autosomal recessive cerebellar ataxias. And so here the people involved in the consortium, so myself on the animal or the preclinical part, Maciej Sinowski's on the clinical part, Francesca Malteca, who's also more on the preclinical part, and Bernard Bray, who's doing both preclinical and clinical aspects. And of course, this is in close association also with ATAXIA and the DHAG and the ATAXIA Charleroi Foundation in Canada. And so, and of course, we have a bioinformatician, Bianca Haberman, who's with us to help us go through some of the large data set that we're going to be generating. So the idea of this project, we're taking two emblematic recessive ataxia disease, and so we're taking RSAX. Ooh, you can't see the colors are really bad. We're taking RSAX, disease in which there's very good animal models and cell models, and we're also taking COC8A ataxia, in which we have developed both also very good mouse models and cell models. And so the idea is to use these two ataxias to do some preclinical research, and so we're going to be doing some validation in the mice about some repurposing drugs, and some of the drugs are common to both disease. We're also, Bernard Bray has did a large drug screening for new compounds, and so we can then validate these new pharmacological compounds in the mouse and cell models. And then we are also developing gene therapy. So that's the preclinical part of it is really to try to develop new therapeutic approach. And at the same time, we're having another aspect, which is biomarkers. So you heard from Paola about the importance of developing biomarkers. And we're going to be using both fluids from humans, but also iPS cells, so these cells, so iPS cell models and then fluids from mouse, to be able to find biomarkers and to see whether some of these biomarkers are corrected in the mouse by the treatment that we're developing, to see if these could be biomarkers to be able to evaluate treatment in the humans. So that's a project overview. I don't have time to go into the details of all the aims of this project, but it's a large project in trying to see if we can be able to manage to be able to find both new therapeutic approach and biomarkers that will responding to therapies in the future. And maybe finally, what I can just point out to what we're also doing with Bianca. This is part of the European project, but it's actually larger than the European project. And we're trying to generate what we were calling an ATAXIA Explorer platform. And so she's a biocomputing person, and her interest is actually to be able to, as you may know, researchers, whether they're preclinical researchers or clinical researchers, we're generating a lot of omics data, all these omics data we're generating transcriptomics, proteomics, metabolomics, imaging. So we're generating a huge amount of data and we actually don't really know how to use them, and we don't even know how to make these available. And so Bianca's idea, and she's already done this for the mitochondria omics, but Bianca's idea is to try to collect all the omics data that are out there in the web from all the different ATAXIA researchers, and to put them together in a single platform, which will be a web-based, available to everybody platform, to be then able to go through and go and ask specific questions that we want to ask in this omic data to see, is there a common aspect between two different ataxias or not? Can we find a common pathway? Can we find specific pathways? And so this is also something that's important to be able to gather all the data together. And so hopefully this ATAXIA Explorer will also be useful for other labs. With that, I'd just like to, of course, thank the funding and then, of course, the people in my lab and you for your attention. Hope I wasn't too long. Yeah, I'm going to put this here.
It's better. Thank you so much. This was very interesting, and now I'm delighted to introduce Dr. Claire Ivancek, thank you so much, who is telling us about a very interesting and very important topic, and yeah, it's just love.
So thank you very much. So I would like first to thank the organizers to have invited me to this wonderful place, a wonderful conference. I'm Claire Ivancek. I'm working in the French Reference Center for Rare Neurological Diseases with Professor Alexandra Dur, that you probably may know. So you must be wondering, why is she bringing this subject? Yes, I will be talking about a difficult subject about end-of-life discussions with patients and caregivers affected by neurogenetic diseases. So it's true that it is a difficult subject, and our aim is to find a cure, but at the same time in the clinics we do the care of the patients, and we're confronted to difficult moments sometimes, and we found that it was also our responsibility to do a research on this difficult subject, because after all we are responsible for the patient. But before I go deep into the subject, just a quick word about legislation, about French legislation, because of course it is not the same in all the countries. But in France, advanced directives have been created in 2005. So what are advanced directives? Probably you may know about it. French citizens may write their advanced directives and designate a surrogate decision maker to assert one's values and choices regarding end-of-life medical decisions in the event that they can no longer express themselves. And in 2016, this legislation has been reinforced, giving unlimited validity to advanced directive and a constraining aspect regarding medical decision. Plus, creating a new right for profound sedation for critically ill patients. And now, actually, in France, the law is evolving. There has been a huge citizen consultation about end-of-life care, and it will probably lead to evolution of our legislation. there is a slowly rising adhesion of French population to this legal device. Within 2021, 18% of French citizens over 50 years old who had written their advance directives. So it is not a lot, but it is slowly arising. And theoretically, patients with neurogenetic diseases like Huntington disease, which is not ataxia, it is another disease that you probably may have, giving cognitive or motor symptoms, or also sometimes psychiatric, but also polyglutamine dominant cerebellar ataxia, which I refer to as SCARS, or Friedreich ataxia, could be interested, could be concerned by this legal device, and theoretically, persons affected by this disease could be prone to discuss about end-of-life issues because of future communication disorders, and also because of the genetic components, which could be a paradigmatic context to anticipate future decisions if you have other persons in your family that you took care of, if you underwent a pre-symptomatic test, if you underwent a prenatal test, for example, for dominant diseases. However, in the daily clinic, this is not what we observe. Advance directives are very rarely written. Surrogates are rarely nominated. There are probably no or few anticipated discussions, and we encounter, as probably a lot of centers, difficulties in addressing end-of-life issues in the medical management. So there seems to be a contradiction between a rational hypothesis, yes, it is interesting, and a daily practice, which is why we addressed a lot of questions. What do patients and caregivers know about this legal disposition? Do they write advance directives? Do they discuss? What are their representations of advance directives and anticipated discussions? What are their expectations? And what would be the determinants that would favor or refrain those discussions? We feel that those questions are important. Why? For the holistic care of our patients, and also to better sustain the caregivers who play a very important role in patients' care. And of course, it is very important societal questions regarding respect of autonomy in situation of vulnerability. And finally, there is insufficient data, insufficient research regarding rare neurological diseases. There are some data, but there are few data, and it is mainly American-Canadian research. So due to differences of law, differences in health systems, we felt we needed to do a French research, which purpose would be to increase knowledge about end-of-life concerns for patients and their primary caregivers affected by Huntington disease, SCARS, and Friedreich ataxia. So this is the Diragen study that I will present to you a few results. It was led by Anne-Claire Dorsemont, who is a researcher psychologist, and I was the principal investigator. So about the method, it was a single-site observational prospective mixed method study, and we proposed the Diragen study to every eligible patient after medical consultation in the clinic. We thought it was very important to avoid biases of inclusion, to be very, very prospective in the inclusion. So we proposed the study to adult patients regularly followed for Huntington disease, Friedreich ataxia, or polyglutamine SCAR, and the patient could designate or not a caregiver that could also participate to the study. 7 days later, all patients filled a questionnaire that assessed global severity of the disease, cognitive, signs, emotional scales, quality of life, and caregiver burden. 7 days later, they filled a specific questionnaire that was designed for the study with the psychologist, assessing demographical data about pathology, surroundings, knowledge of legal device, advance directives writing, nomination of surrogate, previous experiences of discussions, and ideal discussion. And finally, 1 month later, a subset of patients had some semi-directed interview with the psychologist of the study. So now I will present you the result. First result is inclusion rate. So we proposed the study to 237 patients, and finally we included, on the whole, 34% of eligible patients, which could be varying regarding pathology. The most included patients were SCA. There were 47% of SCAs that participated, followed by HD, and subsets of patients who participated less were Friedreich ataxia patients, 27% of participation. I will be quick for the socio-demographic characteristic, mainly to tell you that mean age of participants, caregivers and patients was around 50 years old. Most of them were educated patients and had spiritual support. Most of them had primary caregiver, 74%, who participated to the study at around a rate of 50%, and mainly the caregivers were spouses. Briefly, participants were not anxious, not depressed, and they assessed quite a good quality of life on standard scales. About caregiver burden, it was assessed as light to moderate by the caregiver. So if we look more deeply into patients' clinical characteristics, we see that as expected, Friedreich ataxia patients were younger than other groups of patients. Mean age of FA patients was 45 years old, and as expected, they had a longer disease duration of around 20 years of duration. Huntington patients and SCAs were aged of around 50 years, 56, 58, and Huntington disease had a moderate cognitive impairment compared to other patients. So now I arrive in the deeper results of the study. Knowledge and use of advanced directive legislation. So here I present the results of patients and caregivers pooled all together because they answered in the same way to these questions. And we compared the results with a poll that was performed in French population in 2021, which was actually really very good luck for us because it's the same year as the Diragen study. So when we asked the participants, do you know about the law allowing advanced directives in France, we were quite surprised to see that only 16% of our participants assessed that, yes, they knew exactly what it is, compared to 30% of general population. About advanced directives, in the same way, there were only 7% of our participants who had written advanced directives compared to 18% of general population. To the question, have you ever designated a surrogate decision maker, there were 28% saying yes in our cohort versus 44% in the general population. So one first quite surprising result of Diragen study was that proportion who knew precisely about advanced directive legislation was lower in Diragen study participants than in the general population. If we look more deeply into the very few who had written advanced directives, we see that HD patients were more represented with 13% of AD reduction, and their caregivers had a higher implication in the study, 90%, comparing to other groups. For example, SCA patients had the lowest rate of AD advanced directives written, 3%, with around 60% of participation of caregivers. So the second conclusion could be that HD patients were more prone to write advanced directives compared to ataxic patients, and they had higher implication of caregivers in these subjects. So now if we look into the big majority of persons who had not written advanced directives, We wondered if they have had previous discussions about these difficult subjects, and we found that 70% of these persons had never discussed of those subjects. When we asked, have you ever wanted to discuss, 58% of the subset said they wouldn't have wanted to discuss, with a proportion of them saying that they didn't previously think about these questions. So this is another conclusion. Not only they haven't written advanced directives and they are not well informed, but most of them who have not written have not had previous discussions, and for a majority, they says they would not have wanted to discuss previous to the study. So a big part of the questionnaire was also dedicated to theoretically the ideal condition for a discussion. And we answered with whom to discuss and on whose initiative. Mainly participants answered that it was a priority to be able to discuss with personal surroundings, and in priority spouse at the patient's initiative. 40% and also answered that it was important to discuss with healthcare professional, in priority general practitioner, more than, for example, specialized neurologist or neurogenetician. When to discuss, so this is probably the most difficult question. There was no consensus to this question. The only consensus was that they wouldn't want to discuss at the diagnosis period, but then they could answer during stability period, after a worsening, and finally, that the answer was at no specific moment judged pertinent by the physician, or when the patient is able to sustain the conversation. These were the answers to the open question in the questionnaire. We will now go into the representation of advanced directives within the couple. So we interviewed 15 couples, 5 couples with HD, so patient, HD, and caregiver, 5 SCA, and 5 Friedreich ataxia couples. And when we asked them to give 3 words that come to mind when thinking of advanced directives, we were struck by the very important discrepancy between answers of patients and caregivers. Patients saying that the 3 words are death, end, and life in this order of importance, whereas for caregivers, the words will be relief, respect, choice. So we see that the answers of patients are very concrete, directly related to their own death, whereas for caregivers, it's more a matter of patients' autonomy and freedom of choice. So in conclusion, we concluded that being affected by neurogenetic disease like SCA, Friedreich, or Huntington's does not seem to prompt people to think about end-of-life planning. So this was not expected. We tried to find determinants that would favor a process of anticipated decision-making, and as HD patient had higher implication, we thought that maybe a prognosis of future cognitive or behavioral impairment would favor a process of anticipated decision-making. Also, the qualitative analysis of verbatim of patients, which I didn't show you here, tells us that there are more interesting determinants like previous experiences in family history, personal predisposition to anticipation, trust and link to the caregivers and physicians, willingness to preserve the close ones. Of course, this study has limits because it is a monocentric study, it is a French study, and it may not be generalizable to other population of patients and caregivers with other diseases, other countries. Also, we possibly have a socio-cultural bias for included population because they were mainly highly educated persons. So about the perspectives and discussion with the audience, in the management, what do we do now that we didn't do before? Well, probably we take even more caution to address this question with persons, patients and their caregiver. there's an obvious need of support to address these questions. Maybe this is not for everyone. And also, we discovered that there's a very important need of support for patient-caregiver discussion regarding the important discrepancies of representation that they have of this subject. On a personal point of view, this study made me change the way I see advanced directives and advanced discussions. Possibly, they are interesting not to decide in advance for a future decision, but to guide future decisions, because anyway, those decisions will occur in a different and predictable context. So I'd like to thank you for your attention. Also, all the co-investigators of the study, clinical research associates, and the clinicians who actively participated, and the partners, among which the Patient Association, AFAF and CSC, and Roche, who funded the study. Thank you for your attention.
Yeah, thank you so much for this interesting results and presentation of the study. Are there any questions to our speakers at the panel? Yes, would you?
Thank you all for excellent talks. A question for you, Claire. The comparison between the patient groups with the controls. When you say controls, were they matched for age?
Yes, they were matched. They were citizens over 50 years. So it was globally matched with the global age of our population. It was the best match that we could have. Overall, they were 50 years old of age. So it's kind of a match.
And one other question, if I may. Did you approach at all the subject of brain donation? Because, of course, that's another issue that we often have to discuss with patients. And immediately when you mention brain donation, people think that, well, that's the end, you know.
No, not at all. We didn't approach this subject, but it's true that it is related.
I want to live with all you 6 guys for a week, and I have a lot of questions, but right now they are gone. But tomorrow they all pop up, or the day after tomorrow, so I'll find you.
We will be there.
This is a question for Professor Junti. Is NFL the biomarker that you talked about? Is it used in trials at all yet?
Not yet, and this is something that we really need to look at carefully. Yeah, the things that is now emerging as well from the Huntington world is that it's also quite tricky to compare different laboratories. So it needs to be centralized, and everything needs to be really have SOPs in how you take the samples, because it could vary even from months to months. And this is key, obviously, if you have a year trial. And this is, something that we need to bear in mind.
Yeah. So would you say that in order for it to be used in a trial, that would be the next bit of work that needs to be done before it's ready?
But definitely for SCA3 is something that, as I also demonstrated in Friedreich, is something that we need to incorporate in the clinical trial design.
Thanks.
Yeah.
Helen, your work is amazing. I've been watching your mouse experiments for years, and some dramatic improvements following gene therapy. I guess the question that everybody asks, both in terms of family and patients affected and clinicians, is what do you think is the trajectory of achieving a gene therapy in humans in terms of time scale?
So that's a very hard question. So as I said, for the cardiac part, it's actually there's two clinical trials ongoing that are currently recruiting and that have recruited a few patients. For the neurological aspect, it's being more difficult because there's been quite a few studies in non-human primates that have showed some toxicity effect on, particularly some toxicity on the dorsal root ganglia. And of course, since this is one of the primary sites of affection also in FA, it's kind of slowing down people. So there's been that issue, and then the other issue has been the amount of vector that has to be done and how to deliver the vector, because do you want to deliver a vector through intrathecal injection so that you need less vector, but then you don't target as much, or do you do an IV, which then would huge amount of vectors and then huge amount of toxicity problems maybe in the liver and things like that. So those are the issues that are being addressed by many pharma companies and many academic sites, and actually not only for FA. I don't anticipate that there's going to be really any trial before at least 2025, but that's my gut feeling. Obviously, these trials are being conducted by pharma, and some of them are by clinical investigators. But if you listen to me or to any of these pharmas 3 years ago, we would say it's in 2 years from now. We did not anticipate at all. So but I think that it's encouraging because it's advancing. But I think that we need to be careful, and this is why it's also important to develop other approaches, and this is also why the pharmacological approaches are important, because at the end it's going to be a cocktail. I mean, gene therapy is not going to be there for everybody. It won't answer all the questions and all the aspects. So, it's not because gene therapy seems like this miraculous thing, but it's important to continue on the small molecules approaches also.
I would like to pose a question to Claire. I deal with many patients, as and it's always a very difficult subject to start. However, we have as well in the UK power of attorney that it's pushed us a little bit in discussing this issue and this subject, and I found it once that you establish a good connection, it's much easier. And as neurologists, I think that we should start this conversation earlier, and not only the neurologists but also the specialist ATAXIA nurse that really has an important role, because our nurses sometimes goes in the house of the patients, so they have much more exposure to the family, to the caregivers, and I think it is something that came out from your survey very, very clearly. Perhaps we should as well review the way that we are dealing with the patients. As Andreas said, I created the ATAXIA centers where, patient is a kind of a hub for a patient for all sorts of issues, even social issues. So it's a place where you can discuss many different situations. As the UK, I would say that we are very lucky in the psychology. The psychologists are really gold dust for us, unfortunately. But in an ideal world, obviously sitting with us is the psychologists that can help and really start this discussion. Thank you.
Thank you. Yes, it's absolutely true. The study showed that there was a huge need for support for those discussions, and some results that I didn't show when asking the patients if they wanted, for example, a specialized, dedicated consultation. Most of them answered yes. But also we found a lot of ambivalence in the person's answers, in the way saying they would like to discuss, in the other way saying not so much. So at the end of the study, I'm not sure it has to be discussed at any time with any patient. The link has to be for sure established, and we need to be very cautious. In France, the legislation has pushed us a little bit too much to assess these questions anytime to anybody, which is, I think, not so good. So we have to find the right place, right time to assess these questions with our patients, if they want to discuss them, because also a quite high proportion of patients, they want to rely on their physicians, which means us. And we have also to take our responsibilities. So it is a very subtle place to find between, yes, giving autonomy to persons, but also respect those who don't want to discuss, those who want to rely on us.
So I have 3 questions. I'm going to be a little greedy and everyone's going to hate me because they want lunch. But first for Dr. Helene, about the mice. What is the plan if you are not allowed to test on mice anymore?
Well, there's no real plan because we're hoping that we'll be able to continue to test, even though it is getting harder. So part of the plan is these iPS cells, so these cells derived from human, and to make these little organoids, and we can actually make also little organoids from mouse cells. Part of the plans are these lower animal models, and zebrafish, is right now it's a vertebrate, so it's actually very close to a mouse and to a human. This is going to sound really strange to you, but we are able to do a lot of testing, and the cerebellum, for example, in the zebrafish is conserved, and so there's actually Purkinje cells, there's the layers, so we can use, and right now the zebrafish, there is ethical aspect, but there's not this frame of being afraid of treating, using animals for the wrong reasons. So right now, so going to lower animals, going to little flies, going to the little worms, the C. elegans, but those are not satisfying solutions. I mean, really what I think is important for us is to have our, and towards the population, because it's the population that is screaming against, that's screaming for animal rights, because they see what's going on in butcheries or even what people do at home with their own animals, and then they don't realize that when we work on animals and when we do experiments on animals, it's actually extremely well ethically oriented, and so we can't do anything. And the mice have to be anesthetized. We need to give painkiller to the mice, like we give painkiller to humans. So but there is this push. But we're hoping that we'll still show that we're doing this in the correct ways, and then we'll still be able to do it, because for our preclinical work, I mean, this is going to be important. You can't do all drug development only in vitro. I mean, so.
Thank you. My second question is for Professor Paula. I don't understand the science completely, so this is going to be really stupid, but is there a reason why the levels were higher in symptoms than afterwards when they were symptomatic?
Yeah, so we were still thinking, and we need really, first of all, increase the number of samples for CSF, because it has been very, very discreet, the number of samples, for the difficulties the patient have in donating the CSF. So we're still watching, and once again, animal models, mice, has been proved very helpful. And so the work from Pereira and University of Coimbra has shown and mirroring basically the behavior that we have in patients. So yeah, but the question is at the moment we still don't have an answer. Maybe it's a compensatory effect. We don't know. It's definitely is at the beginning. So it's an interesting observation, obviously.
Yeah.
And because it can be used as conversion biomarkers.
Amazing, thank you.
Yeah.
My last question is for Dr. Clare about end of life. At the moment, in the community, in the ATAXIA UK community, I'm working on providing this kind of information easily. And so it would easily sit on the website for somebody who is ready to read it, to come in and be receptive to the information they hear. Is that something you would recommend doing? Just making it, I think the topic is so taboo, but if we started to talk about it, it would be easier and it would be something that was just as much as living. We talk about the end and what we do when we get there. So my question is, do you think it is worth exploring and making it more readily available, and would that then take away the taboo around the topic?
My opinion on this question changed during the study. My pre-hypothesis would be to answer yes. At the end of the study, I'm not totally sure. I think what we should provide to persons is the fact to tell them, assure them that we are open to the discussions, yes, as physicians or nurses or psychologists, about giving pieces of information on internet. Actually, in the questionnaire, we asked this question. So participants answered that they would like information, brochures. I think it's difficult to interpret these questions because in the study it was very reassuring for them because they were face-to-face with psychologists. So as to transfer these answers to general population and patients and caregivers seeing this on the website, I'm not totally sure. I think that we should tell them that there is a place to discuss, yes, and that the subject can be brought on the table. I'm not sure that we should give tons of elements about these questions. So I don't have the answer really. I think we have to be cautious. You can give elements about the law, the rights, patients' rights, but will it achieve the goal? I'm not sure, because in the end, what they need is to discuss, and they need to be in front of someone. So yes, possibly it's better than nothing, but still, you need to be quite cautious when you put those documents accessible to everyone.
Okay. Thank you very much.
Thanks for the nice presentation. I have a question about the gene therapy. By translating that in humans, do you anticipate an age barrier on that in terms of immune reactivity or viral load?
So I think the age barrier, there's going to be a barrier that is whether you've been already exposed or not to AAV, because AAV is actually a naturally present virus in the system. The age barrier, more than being an immunity answer, would be actually when's the right time to treat. You know, with gene therapy, of course, if the cells are gone, you cannot treat them. So we need to be able to treat when the cells are dysfunctioning but still there. And so that's where the age barrier. So you need to treat when early enough so that you can still hope to stabilize the cells that are there and prevent them from
exactly.
disappearing.
Yes, we see that in SMA. We need to treat as soon as possible. Can you bypass the viral load thing by intrathecal injections?
So that would be, so that actually most of the programs that are ongoing are looking at that, are doing either by intrathecal injection or by intracerebral injection to bypass the viral load that would be necessary. So yeah, that would be one way. Just one thing that's an advantage in FA is because FA is low level of frataxin, so we're not going to get an immune, or we don't expect to have an immune response against the frataxin gene, because all patients still have an amount of, so there won't be that reaction that can occur in many of the gene therapy settings where they've never seen the protein before. But yeah, so reducing the viral load would be through intrathecal.
Thank you for your question. Thank you for the flash talks.
On Zoom, but we are going to forward them to you since we are, let's say, 40 minutes out of the schedule, and we have some people that they are connected to give their presentations afterwards. That means that it was an excellent panel, and thank you again.